Texas Lutheran University.
So today we have Michael Penrose, and Michael graduated from Grace Preparatory in New Braunfels, and you've been here all 4 years, yes? Yeah. Okay. I didn't have him in Gen Chem, and neither did you, so we
Yeah, Dr. Toledo with me.
And you still decided to stay?
Impressive. I'm particularly proud because he was a USDA intern last year, and with no information, because I didn't have any, he basically got on a plane and went to DC and went to intern. It was completely a step into the wild blue yonder, but he did great, and they tell me he was amazing.
We only bailed him out once, right? Just once?
Only once.
Yeah.
Well, I couldn't find the job at the USDA, so his manager at Wendy's really liked him.
Anyway, today he's talking about the inhibition of complex 1 of the electron transport chain of Rotenone.
Yep, well,
Yeah, I know.
close enough.
I'm going to just go choke on that and turn it over to Michael.
Okay. So as you just heard, my research was about the inhibition of complex 1 by Rotenone.
Do you have to go back and click it on the PowerPoint?
Yeah.
It went out. So it starts in the mitochondria, which is the powerhouse of the cell, providing all the energy for the cell. Within the mitochondria, you'll find the electron transport chain, which is a metabolic portion of the cellular respiration, which produces most of the energy for the mitochondria. With that, you'll find complex 1, which is what this is about, and it's responsible for a large amount of that energy. And it is also the source of reactive oxygen species in the mitochondria, so it is very important that we make sure nothing happens to it. But of course, things can happen like in the inhibition with Rotenone. Rotenone is a model inhibitor for complex 1, being that it provides a demonstration for all the inhibitors in general. And we have actually learned a lot about complex 1 through studying Rotenone. Rotenone is commercially available, so it's easily findable both as an insecticide and a pesticide, but it is especially effective as a pesticide, which is a fish killer, because it could get in their bloodstream through the gills. As you can see here, it was super effective. So, but the problem is it's not selective, so it will kill any fish. So you would have to actually reintroduce the desired fish back into the system after Rotenone was decomposed in the water. So to understand the inhibition, we first have to cover the ETC for complex 1. So the electron transport chain is located in the mitochondria, which is found in the cell, as you can see by this picture on the left. If you can see it, the organelle mitochondria is in there. And in the inner membrane of the mitochondria, which is this part right here, you can see that is where the electron transport chain is located. So complex 1 is very large, by far the largest complex of the ATC. It's between 40 and 50 subunits, but we could it's easier to split it into 2 major parts, which is the hydrophilic arm, the only portion of it to be seen by X-ray crystallography is the location of most of the interactions, whereas the hydrophobic region is located hidden inside the membrane and is the site of the final interaction. And there's also 8 FeS sites, which you see here. 2 of them are Fe2S2, and the other 6 are Fe4S4. So the main function of complex 1 is to pump protons in the form of H+ across the membrane, which is coupled to ATP production. This uses a series of oxidation-reduction reactions to pass electrons through the complex into future parts of the ETC, while also controlling, again, production of reactive oxygen species, or OOS, as they are often called. So the electrons start at nicotinamide adenine dinucleotide, luckily called NADH. And then it passes it to FMN, which then passes it through the surface centers and out coenzyme Q. So NADH is produced earlier in different metabolic pathways to be used here, and its kind of scary structure is shown down here. But of course, the only important part is this little portion there. The rest of it we could just simplify it and call it R, as shown here. Which is very, very good. So NADH is oxidized to NAD+. What happens is the hydrogen, it loses a hydrogen and 2 electrons and forms this structure on the right, which has a positive charge on the nitrogen. NAD+ is reused in earlier metabolic pathways to form NADH by becoming an electron acceptor. And by forming NADH, it's recycled. Isn't it great how metabolism uses recycling? It's a good example for everyone. So FMN, also known as flavin mononucleotide, I'll never refer to it as that, is widely used in oxidoreductases. Oxidoreductases are enzymes like complex 1 that catalyze the transfer of electrons. It works well as an electron carrier. It is easily produced from riboflavin, which is a vitamin, also known as vitamin B2, which is found in many food groups, so it is easy to get. And here, its structure is on the right, but of course this is the only important part that takes place in the interaction. And the rest of it we can just, again, simplify as R. So FMN actually takes 2 electrons, so you can use 2 NADHs to give the electrons to FMN. And it is a stronger oxidizer than NAD+. And the reduction was shown on the right. As you can see here, 2 hydrogens are added to 2 of the nitrogens, which are here and here. So once again, there's 2 types of iron sulfur clusters, Fe2S2 and Fe4S4. And it is important to note that N2 is an Fe4S4 cluster. The structure of both of them could be shown at the bottom. But Fe2S2 has 2 sulfides and 2 irons, as well as 4 cysteine groups. So I think it's important that you know what cysteine looks like for this. So I added it here on the right. And the S, or the sulfur from the cysteine, actually binds to the iron in the system. Fe4S4 has the same amount of cysteine residues, but it also has 4 sulfides and 4 irons. So now that it has reached the N2, or the last cluster, there are 2 ways that the electrons can go. They can either go to coenzyme Q if it's present, but if it's not present, they will leak to oxygen and form superoxide radicals. Coenzyme Q, also called ubiquinone, or for short Q, is the final electron acceptor of complex 1. It passes the electrons to later parts of the ETC. And its structure is shown on the right, and it's actually kind of scary, but once again, you just use this part here. What happens are the 2 double-bonded hydrogens are reduced to form an alcohol group in both spots. And once again, once it is reduced and goes to the next part of the electron transport chain, it could then donate its electrons and go back to complex 1 to be reused and recycled in complex. So there are 3 major, very important points of kinetics in complex 1, and they are the favorability of the oxidation of NADH versus the synthesis of ATP, the iron-sulfur cluster midpoint potentials, and the CoQ enzyme kinetics. So ATP, also known as adenosine triphosphate, is the molecule that carries energy. It is produced through phosphorylation of ATP, but it was kind of unfavorable. The reason it carries energy is because, if you see by the molecule on the right, has 3 phosphates, but ADP only has 2. So when hydrolyzed, one of the phosphate groups is broken off, releasing a whole bunch of energy. Using that term loosely. Okay. I don't know if you can see this one on this computer. So once again, the ATP formation is pretty unfavorable, as shown here by, I used Gibbs free energy, or delta G, where a negative delta G means that the products are favored, and a positive delta G means that the reactants are favored. And here, the oxidation of NADH has a negative delta G of 53.6 compared to the positive delta G of the phosphorylation of ADP to ATP, which is 7.3 kcal per mole. But of course, since NADH has a higher negative value than ATP has a positive value, it releases enough energy to cause the reaction to proceed. So ADP will become ATP. And FMN is both reduced and oxidized in this system, so since they contradict each other, they balance out and it doesn't end up in the final equation as shown here. So as you see here, NADH is oxidized and then coenzyme Q is reduced, but there are also 4 hydrogens that are moved from the matrix to the intermembrane space as shown by this picture. So the hydrogens are moved from the matrix down here up to the intermembrane space up here through the complex and by coenzyme Q. So once again, there are 2 types of iron silver clusters, and they each have different midpoint potentials. Midpoint potentials just kind of identify whether they're going to be better for reducing or oxidizing. So lower midpoint, a negative midpoint potential means that the molecule would rather be oxidized, where if they have a positive midpoint potential, they'd rather be reduced. Here you can see that Fe2S2 has lower midpoint potential than that of Fe4S4s, having a midpoint potential of a negative 370 millivolts. And Fe4 has S4 can have anywhere between negative 150 and negative 50 millivolts. The N2 cluster is known to have the highest midpoint potential out of these, and so meaning it is less negative, so is more likely to hold on to its electrons. And being the glory hog that it is, it would just love to do that, so it could be the one to hand off the coenzyme Q hand off the electrons to coenzyme Q. So coenzyme Q is it has a high diffusion rate, meaning that it's a good electron carrier for this case because it can easily move across the membrane and carry it to the other parts of the electron transport chain. It also has a high rate of electron transfer, but a low rate of attachment, giving it an unfavorability for its interaction. But of course it does work, so there has to be something driving its interaction. So as far as I know, there are 2 possible driving forces. The first one being that the coenzyme Q actually interacts as a pool, where some of the coenzyme Q is bound to the membrane, but the rest of it's actually free floating around complex 1, so that way it can interact as a group rather than just one in itself. And here is the pull equation on the left. The other one, for people who don't believe in the pull equation, is that just that NADH oxidation is favorable enough to actually cause the interaction to occur. So the pull equation. In the pull equation, V is rate, meaning that V reduced is the rate of reduction of coenzyme Q, and V oxidized is the rate of oxidation of coenzyme QH2. And V observed is the metabolic flux, also known as the maximum turnover rate, which is the rate at which these oxidation or reduction reactions occur. And I reuse this image here because you can see how N2 here is reacting with the different coenzyme Q enzymes here, or coenzyme Qs here, showing that it interacts as a pool rather than individually. But of course, the pool equation does require a specific set of conditions. And that is that it requires a fully saturated solution for both the oxidation and reduction components, meaning it needs excess coenzyme Q and coenzyme QH2 in order for it to be used. It also assumes a steady state of electron transfer. If these conditions are not met, then you can correct it by feeding the pool equation into Michaelis-Menten equation, which is like the best equation in the world. For you to know it. So this takes into account the total Q rather than each component individually. But in Michaelis-Menten equation, V is rate, V max is the maximum velocity, S is the substrate concentration, and Km is the substrate concentration at 1/2 V max. And it determines how well the reaction takes place. Now we can talk about rotenone, finally. So, rotenone is easily decomposed in the presence of oxygen and sunlight, and it's very insoluble in water. So it would rather interact with polar molecules in the water, other non-polar molecules are in the water, rather than interacting with water itself. Here is the structure of rotenone, which will come in handy very soon. So the breakdown of rotenone. Rotenone, again, is highly sensitive to light and oxygen. So that means the major path of breakdown, the major path that it breaks down is through photochemical oxidation, by which it uses light to remove electrons and allowing it to be oxidized by oxygen. Well, this forms rotenone, which is a major intermediate and is also much, much less active than natural rotenone. It also can be hydrolyzed in water in the presence of H plus and H2O. In this case, side chains like the methanol groups over here, or the methanol groups can be hydrolyzed. But this is slow. This is a slower decomposition, still allowing for it to interact with its fish friends, while also being able to be removed from the system over the course of a few days. So the inhibition works because rotenone is specific to complex 1 and has a higher binding affinity than that of coenzyme Q, meaning it can just perfectly just come in there and block the transfer from NADH to coenzyme Q at the N2 site. But since the structure of rotenone does not match coenzyme Q like a good inhibitor should, it is believed that either only part of it binds or the shape causes it to bind. And of course, the two major problems caused by the inhibition are the decrease of ATP production and the formation of superoxide radicals. So exactly what kind of inhibitor is it? There are two major classes of inhibitors that are specific to complex 1. There are class A inhibitors and class B inhibitors. Class A inhibitors increase superoxide formation while also decreasing ATP production. But class B inhibitors decrease ATP synthesis as well, but also completely block the formation of superoxides. An example of this is stigmatellin, which I have shown here for completion purposes. And of course, there are three types of general inhibitors. They're competitive, non-competitive, and uncompetitive, which I will show here. Competitive, like rotenone, in competitive inhibition, like rotenone, the inhibitor actually goes to the site and binds directly to it and blocks the substrate from binding. But of course, in this case, it has to be similar to the substrate and can be removed at high substrate concentrations. Uncompetitive inhibition instead will bind to a separate area on the enzyme, which actually causes it to change shape and will block the substrate binding due to the change in shape. An example of this is cyanide, which does affect complex 1 as well as other parts of cellular respiration. Uncompetitive inhibition is when the substrate binds to the enzyme, but then the inhibitor binds afterwards and blocks products and the substrate from leaving. There's no actual uncompetitive inhibition in complex 1, but an example of this would be lithium in the brain. I hope you can see the picture. Loss of ATP means lack of ATP. So it is important that we do not lose ATP because it is the energy of the cell. Which in the loss of ATP also causes dysregulation in production of many metabolites, And when you don't have the metabolites, it creates problems for other metabolic processes as well. And you don't want to be this cell here, because he believes that he can create he can make more mitochondria to solve his energy problem. And mitochondria replicate independently of the cell. The cell cannot make its own mitochondria, so it can't save itself from the low energy problem. So complex 1, once again, is a major site of superoxide production. And it is known that as rotenone inhibition increases, so should the superoxide, so should a few ROS species, which are the two types of ROS that are produced from the inhibition are superoxide species, superoxide radicals, and hydrogen peroxide. And the increase of radical formation causes both oxidative damage and other negative impacts on mitochondrial functions. So here are the possible ROS products, where the two important ones here are going to be hydrogen peroxide and superoxide anion. And the cool part is that superoxide anion actually produces hydrogen peroxide in the same amount of quantity. I guess that's how I'm saying it. So here's how the superoxide radical is formed. Originally, both oxygens have one lone pair, but once an electron is introduced, one of the oxygens will take the electron, and the molecule will become negatively charged and highly reactive. So, superoxide production. Superoxide radicals and other ROS, mainly hydrogen peroxide, are produced at the iron silver clusters. This happens when coenzyme Q cannot take the electrons from the site, so they remain at the N2 site, and electron leakage to oxygen increases. This causes superoxide cell toxicity, which is a result of overproduction of superoxide, and this leads to oxidative stress caused by an imbalance between the formation of superoxides and the cell's ability to neutralize them. This hinders the cell's ability to perform natural redox regulated functions. And then the oxidation by the radicals can also cause modification in proteins, breaks in DNA, and eventually leads to cell death. So there are two major features that impact rotenone binding. The first one is the shape of rotenone, which can be determined by the configuration and the conformation, which was shown. The effect of that was shown by an experiment that compared natural rotenone versus modified rotenones. And the second one is concentration, on which two experiments were done, one against cellular respiration activity and the other against hydrogen peroxide production. So conformation is a single rotation around a sigma bond, and this does not require bond breaking and does not change the chemical composition, whereas configuration requires bond breaking and it does change the chemical formula. So you can see this. So the experiment measured the activity of natural rotenone versus its two stereoisomers, 5' alpha epirotenone and 5' beta epirotenone. 5' alpha epirotenone, the only change was on the side alkyl group over here, where it was changed from the up position to the down position. This proved to be very effective because alpha epirotenone only has 10% as active as natural rotenone. And 5' beta epirotenone only had the change in hydrogens where both were pointing up, and now they're both pointing down. And what happened was it was only 0.1% as active. And then synthetic rotenones are the ones that are used. They often use configurational changes to see how that works, and those are even less effective than the stereoisomers shown here. So the other important aspect is concentration. Here it was shown that as rotenone in concentration increased, the percent of cellular respiration decreased, also meaning the percent of ATP produced also decreased. And the inhibition was detectable at concentrations as low as 10 nanomolars. But also, if you notice by the graph here, that the slope as concentration increased also decreased, and the slope as the inhibition rate are also, you can consider it the binding affinity. So as it increases, the more you add on, the more the next rotenone, the less likely the next rotenone is to bind to complex 1. But it leveled off at 500 nanomolar, showing a saturation at that point. So here, hydrogen peroxide production was measured against the concentration of rotenone. This was done because we were concerned about superoxide production, but since superoxides are pretty hard to measure, and you know that the formation of hydrogen peroxide should be about equivalent to hydrogen peroxide to the superoxide production, you could actually measure hydrogen peroxide to measure about the amount of superoxide. And it showed a similar graph to as was shown in the cellular respiration inhibition. What happened here was as the concentration increased, so did hydrogen peroxide formation. But just as before, as the slope decreases as you increase, meaning that the rate at which it increases the formation of hydrogen peroxide decrease as you add more and more. And once again, it leveled off at the exact same point of 500 nanomolar. So rotenone poisoning is what occurs when the inhibition becomes hazardous. And once rotenone inhibition is noticed before it gets to this point, you could prevent it by a few ways, but two of them are shown here. The first one is through use of a class B inhibitor, which, as told earlier, completely stops the superoxide formation. So this will help against the superoxide formation, but it doesn't really solve the ATP problem. But there are ways to increase activity of earlier metabolic pathways, so that way they can produce more ATP to try to substitute until the inhibition is removed. Or you can just skip complex 1. Yeast does this really very effectively using an enzyme called ND2, which it takes the electrons and moves them past. But we don't have that. So we just have to deal with the loss of ATP, but that's a problem because the ratio of ATP produced by complex 1 versus the ratio of ATP produced by the rest of the complex is 5:3, meaning complex 1 produces more ATP than the whole rest of the complex does, the whole rest of the ETC. So future research for this topic is to observe the functionality of complex 1, because that will help understand the inhibition by rotenone. Or if you look at the inhibition by rotenone, as has been done before, you could actually understand more about complex 1. But of course, rotenone is very hard to work with due to its unstability in oxygen and sunlight. So the most important thing is to find ways to make it easier to work with. So acknowledgement. First, Dr. Wayne for being my mentor and putting up with this presentation, with this research since last fall. Dr. Bray for, in a way, forcing me to practice my presentation early. In both chemistry and biology departments, because I actually understood the research, which means I may have possibly learned something, which I was surprised to find out. And now that you've heard me drone on about rotenone and complex 1, does anyone have any questions?
For more information, please visit tlu.edu.